Today's session is about metabolic chemistry. And when we're talking about metabolic chemistry today, we're really thinking about metabolism and the processes that are involved in metabolism. So catabolism, which is the breakdown, and anabolism, which is making or building. And we're going to also talk just towards the end about two of the key regulators in glucose metabolism, if you like, fuel metabolism, how we regulate the amount of fuel we have within our bloodstream that is then able to deliver to our tissues. But first of all, we're going to look at the main metabolic processes in the production of ATP. And of course, ATP is the energy form of the body. It's what is used for us to do the mechanical work that we need to be able to do, whether that's pumping ions, or whether it's our smooth muscle contracting, or whether it's our brain thinking and generating those action potentials. It's ATP is the energy source. And metabolism refers to all of the chemical reactions that are taking place in the body. And it's broken down, as I've already said, into two basic processes. You've got anabolism and you've got catabolism. So anabolism is all about making. And so in other words, it's storing energy. And in the process, it is using ATP to produce ADP and phosphate, because that's what ATP comes from. Whereas catabolism, on the other hand, is designed to break down molecules to produce ATP for work, for whatever that work that might be. And this produces heat as a byproduct of catabolism. So in other words, what we're really thinking about, metabolism is providing the energy, the ATP required to maintain homeostasis and perform essential functions. Now, some of those functions, the ATP might be required for breaking down and replacing cells and their components, which we're doing all of the time. It's required for growth and cell division, so we can get bigger, we can make more cells. And for various secretions throughout the body requires ATP. Contraction, if you're thinking about your muscles. Nervous system, action potentials, generation of action potentials, and therefore information transfer throughout the body via the nervous system. So if we look at the little diagram that we've got at the bottom here, and this is just showing a cell and around it the interstitial fluid, because that's the fluid that bathes the cells. And in there, you'll have some nutrients. So we've got amino acids, we've got lipids, and we've got simple sugars, such as fructose and glucose. And they get taken into the cells, and they make up our nutrient pool. Now, we can use ATP in times of when we've got lots and lots of energy, or lots and lots of nutrients, rather. We then want to store it. So that's anabolism. So it uses ATP, and it stores energy. So amino acids as proteins, lipids as triglycerides, simple sugars as glycogen. But it also allows us to maintenance features. It allows us to growth. And as I said, it's making those proteins, those lipids, those carbohydrates, and nucleic acids, so your DNA and your RNA. By contrast, in between meals or when you're having to repair things, you're breaking down, and that's your catabolism. And you're breaking them down back into that nutrient pool of amino acids, of lipids, and simple sugars. And within the cytoplasm of the cell, and in particular when we're thinking about glucose, that can get broken down anaerobically to produce a very small amount of ATP. And under anaerobic conditions, of course, that goes on. Eventually, it would go on to make lactic acid, such as in your anaerobically respiring muscles, skeletal muscle, when you're working very, very hard. Or your red blood cells as well. They only work anaerobically and produce lactic acid. And the byproduct of that is lactic acid, but of course, further down it actually forms pyruvate, another molecule you'll need to know just a little bit about in this session. So pyruvate, so glucose is metabolized down to pyruvate, and that can go on to form your lactic acid, or it can enter the mitochondria. And in the mitochondria, we start to undergo aerobic metabolism, so it's using oxygen. And that produces your CO2, produces a lot more ATP, and therefore that ATP can go on and do all these functions that we've talked about. So movement, contractions, intracellular transport, endocytosis, that's bringing things into the cell, and exocytosis is sending things out from the cell. And as a byproduct, we produce a large amount of heat. So when we're thinking about it, catabolism is what we're focusing right now, and that produces your ATP and your heat. And so your fats and your proteins and your carbs, they're all hydrolyzed or broken down into their building blocks. So we break them down, proteins into amino acids, carbohydrates into monosaccharides, such as your glucose, and your fats into glycerol and fatty acids, because you normally have a glycerol backbone with fatty acids coming off it. Your second stage is conversion to acetyl-CoA, so it's another little intermediate that we'll touch upon later. And then stage 3, that intermediate acetyl-CoA, that can enter the Krebs cycle or the TCA cycle, and we're in the mitochondria at this stage. So when we're in the mitochondria, that's when we're using oxygen, and that's when we produce huge amounts of ATP and carbon dioxide as well. So like I said, oxidation of this acetyl-CoA in the Krebs cycle, that and the electron transport lane, they're all kind of linked together in the mitochondria. They go on to produce your ATP and your carbon dioxide. And we'll come to that later on, but it's important at this stage that you just have a crude understanding of what the processes are called and what's going to happen in each stage. But of course, the whole of metabolism and all of our cells, there needs to be this metabolic integration, and each tissue needs to be provided with usable energy sources to perform specialized functions. Now, all our cells are using this molecule, adenosine triphosphate, so an adenine molecule, ribose, and 3 phosphates. And by breaking one of these phosphates, you release lots of energy that allows us to do stuff. And again, you break another one, it releases energy. But then to make the ATP, that requires energy again. And like I said, all of our cells are using this ATP as an energy source, but they actually get it from different places. They have different molecules that can be used. So the brain, for example, primarily uses glucose as an energy source to produce ATP, though it can produce ketone. It can use ketones to produce ATP. It can't use fats. Okay. Whereas, of course, our heart and our skeletal muscles, they can use fats, in particular our muscles at rest. They're just using fat as an energy source to produce ATP. So I just want you to think just quickly, what are the main functions that are going on in each of these tissues? So what's happening in your muscle? Well, that's quite an easy one, so I'll even do that for you because it's mechanical work, it's contractions. But really, press pause and have a think, what's happening in the liver, the kidneys, and the neurons? Well, of course, in the liver, there's an awful lot of biosynthesis going on. It makes most of our plasma proteins. It makes glycogen. It can make glucose as well, and it can make triglycerides. So it's making an awful lot of things. The kidney, pumping of ions. So there it is. It's pumping sodiums and potassiums and hydrogens and other ions as well. And that takes a vast amount of energy. And neurons, of course, they're generating action potentials so that our nervous system can communicate with itself, within itself. And of course, energy production must meet the various needs that we have at different times depending on which tissue you're in. So right now, if you're not doing very much, your skeletal muscle isn't using very much energy. But at different times, it might need to have lots of ATP for that muscle contraction. So it might be using fat now, but later on, it may go on to be using a lot more glucose because you get a faster release of ATP using glucose. So therefore we have tissues, our tissues express specific enzymes enabling specialized storage, usage and generation of a variety of molecules. So I want you to think again, what are the major energy storage molecules within the body? There are 3. And what are their storage locations in each case? Again, 3. So again, have a think. So hopefully you've got the three places where they're stored. So we have our adipose tissue, our fat cells, and they of course primarily store triglycerides. We all should know that. We have our liver and skeletal muscle. They both store glycogen, and glycogen is just long molecules of glucoses joined together. So it allows a large area of glycogen storage that we can then break down into glucose when we need it. And our skeletal muscle, they store a lot of protein, which can be broken down. During fasting, we need to break down our protein into amino acids, which can get converted to glucose, which then the brain can use as an energy source. For example, if we've run out of our liver glycogen, we need to be getting blood glucose from somewhere, and it's coming from our amino acids. So, what are the 3 main processes when we're thinking about the metabolism of glucose? So the 3 main processes: glycolysis, the citric acid or Krebs cycle, and the electron transport chain. And you need to have a basic understanding of these 3 processes because it forms fundamentally parts of your learning outcomes. So when we're looking at it, here's a nice cell surrounded by our interstitial fluid, and we take up our fatty acids, our glucose, and our amino acids. And we can then store them as triglycerides, glycogen, or proteins, depending on the tissue. But when we're breaking them down, we can break them down into our 3 carbons. So these, glucose in particular, if you think about it, is 6-chain carbon molecules, C6H12O6. So we break it down into 3 carbon chains, into pyruvate, which, if we work anaerobically, can go on to produce lactic acid. So it goes to pyruvate first and then lactic acid. But it only produces a very small amount of ATP, just 2 ATP molecules. And after then, it goes on to enter, this pyruvate enters the mitochondria, and it immediately gets converted to acetyl-CoA. So you remember earlier we talked about acetyl-CoA. Well, the pyruvate's converted to acetyl-CoA and it enters the mitochondria, and there it enters the citric acid cycle, and we now become aerobic. And throughout this whole cycle and the linked electron transport chain, we basically use oxygen, produce CO2, produce water, and produce lots and lots of ATP. So aerobic respiration in the mitochondria generates a further 30, roughly, ATP molecules. So we produce 2 from glucose in the cytosol and a further 30 if we're doing it aerobically in the mitochondria to produce 1 glucose plus 6 oxygens gives 6 CO2s, 6 waters, and 32 molecules of ATP. So, I've already actually touched on this. Which tissues in the body rely solely on anaerobic glycolysis for their energy needs? And what's the end product? So that's my first question. Now, hopefully you should be able to do this one quite easily because I talked about it. And it's your red blood cells, okay, your erythrocytes. And during anaerobic respiration, of course, your skeletal muscle also works anaerobically. And what was the end product of this? It's, of course, lactic acid. And lactic acid is then removed from that cell, enters the blood, returns to the liver, and in the liver it is converted back into glucose again. So quite a nice little useful cycling pathway. So I want you to complete the following table of definitions, telling me the main cell type where it occurs. So I want you to define glycolysis. I want you to define glycogenesis. Remembering what is genesis, what does it mean? Same thing, glycogenolysis. What does lysis mean? And then gluconeogenesis. Again, define each one and then just tell me, or tell yourself, what cell or cell types is this taking place in? So press pause. Okay, so glycolysis. So glycolysis is the anaerobic catabolism of glucose to pyruvate or lactate, producing ATP. So it only produces 2 ATP molecules. It's taking place in the cytoplasm of cells, and it's taking place in every cell that I can think of. Okay, so it's taking place in all of our cells. Generally, it forms pyruvate, okay? But in red blood cells, it forms lactate. In anaerobically working muscles, it forms lactate. But generally, it'll produce pyruvate. So glycogenesis, that is the synthesis of glycogen from glucose. And that takes place mainly in your skeletal muscle, that's what the M stands for, and your liver, which is what the L stands for. Now, during periods of starvation, that glycogen would get broken down, form back into glucose, which is this next process, breakdown of glycogen to glucose. Okay, during starvation, that only really happens in your liver to any large extent, and that glucose then enters the blood. During a fight or flight response, for example, you break down your glycogen to glucose in your skeletal muscle, but it does not enter the blood. It stays within that muscle and is used in that muscle only. So basically, when you make glycogen in the liver, it's broken down to glucose. That glucose can enter the blood and go to wherever it needs to go, often the brain. In the case of the glycogen that's broken down in your muscle, your skeletal muscle, yeah, it's broken down to glucose, but that glucose does not leave the cell. It remains within that skeletal muscle cell and is used by that cell. So it's a very important differential between the two forms of glycogen, if you want to think of it that way. And the last one is gluconeogenesis, and that is the synthesis of glucose from non-carbohydrate substrates. Okay, and we'll talk about that a little bit later. That takes place primarily in the liver. Like I said, the liver does a lot of synthesis. It's synthesizing glycogen, it's synthesizing glucose as well from non-carbohydrate sources, such as amino acids when you need them, for example. So now we're going to go on to the metabolism of lipids. And as you may expect, lipogenesis is the synthesis of lipids. So it's the synthesis of fatty acids and triglycerides. Of course, some fatty acids cannot be synthesized in the body. These are our essential fatty acids. And our essential fatty acids, though we get them, tend to come from plants, and they are required to make some of the phospholipids in our plasma membrane, our cell membranes. Many tissues can use fatty acids as an energy source, so skeletal muscle, cardiac muscle, liver cells. But of course, our brain cannot use fatty acids as an energy source. It relies on glucose at all times as an energy source. So even during prolonged starvation, when the brain is still functioning, obviously, it still needs to get at least 50% of its energy from glucose, and the other 50% during starvation comes from ketones or keto acids. So it can't use fatty acids as an energy source, unlike other tissues. And our triglycerides, they're by far the most abundant lipid in our body. They provide 1.5 times the energy of glucose per carbon molecule. Okay, so per carbon, they produce about 1.5 times more energy. So they're very energy efficient molecule to have. They're very high density and they're longer term energy storage molecules. They're high density, long term energy storage molecules. And so that's lipogenesis. And of course, lipolysis is breaking it down. So within our adipose tissue, your triglycerides are broken down into your glycerol and your free fatty acids. So here's your blood, here's a adipose cell, and here are your triglycerides. And there's basically a little enzyme in there. It's called hormone-sensitive lipase, and that breaks down your triglycerides into fatty acids. The FFA stands for free fatty acids, and your glycerol backbone, which can then go to the liver and be reconverted into glucose. And for example, adrenaline is an example of a hormone that will stimulate this enzyme to release free fatty acids into the blood to be able to use as an energy source, and they get metabolized aerobically. So next we're going to do proteins. So proteins are an interesting one. We make up to 140,000 different proteins. And they all have different functions or subtly different functions from one another, and they're in different cells. So we have different proteins expressed in different cells, even though the DNA in every one of our cells is identical, with the exception of our germ cells, of course. But when consumed in excess, amino acids get converted to glucose, yes they do, but they also get converted to fats. So if you have a very high protein diet, you'll be making a lot of extra fatty acids and triglycerides out of the excess amino acids that you eat. Protein is only really broken down for energy if insufficient supplies of carbohydrate are present. So remember, if you don't have enough carbohydrate in your liver, if you've broken down all your liver glycogen, for example, you still need to feed your brain with glucose. Where does it come from? Well, we start to break down our proteins. We break them down into amino acids, and the amino acids are then converted to glucose, but it produces about the same amount of energy as carbohydrate. That's about 4 calories per gram. I work in calories, so if you want to work in kilojoules, just times it by 4.2. So that's about 16.8, 17 kilojoules. But protein breakdown does produce toxic ammonium ions. And so that ammonium obviously is very toxic. You don't want that in your blood. It does all manner of damage. So it's metabolized to urea in the liver, and you excrete urea in your urine. But of course, extensive protein catabolism, so if you break down too much protein, if you starve for too long, you're breaking down your muscle, you're breaking down your other cell products, so the proteins in other cells as well, it threatens homeostasis because it basically starts to destroy the structural and functional components of cells, because that's what cells are made of. Structurally, yes, they have a lipid membrane, but there are proteins in that membrane doing essential jobs. They have functional roles, just like they do in the cytoplasm. If you're running out of ATP, if you're running out of carbohydrate, you have to make more carbohydrate, and you make it from amino acids. So to do that, you need to break down your proteins. So what I'd like you to do, another little review question, just a fairly simple one. I just want you to define the following. So we've actually talked about it. So define each of these and tell me where it takes place. Lipolysis, obviously catabolism of triglycerides into glycerol and fatty acids, primarily in the adipose tissue, though we do have them stored elsewhere. Lipogenesis, the anabolism of fatty acids and triglycerides, mainly in adipose and liver. Proteolysis, catabolism or breakdown of proteins into amino acids, primarily your skeletal muscle and your liver. and ketogenesis. This is the anabolism of ketones from fatty acids and some amino acids and takes place in your liver. Remember, the brain can use ketones during that starvation phase as an energy source as well as glucose. It always requires glucose. So therefore, what you can see from this, if you're looking at that, like the liver plays an absolutely essential role in regulating our plasma amino acid levels and by its same roles in our glucose levels, because it plays a role in glycogenesis, making glycogen, glycogenolysis, breaking down glycogen, gluconeogenesis, making glucose from non-carbohydrate sources such as our amino acids. So the next bit we're going to talk about in a little bit more detail the processes involved in both catabolism and anabolism of the variety of different biomolecules. So as we've already talked about, glycolysis, that occurs in the cytoplasm of cells. It doesn't require oxygen, and it is an anaerobic process. And so this is where we're going into a little bit of biochemistry. So I'll try and go quite slowly so you can start to understand it, though it is quite complex. So do take your time, and I'll provide a couple of links later on that may help you. So what's happening when we're thinking about the breakdown of glucose within the cytoplasm? What's actually happening? We're trying to create ATP. Now remember, we don't make very much ATP. We just make 2 molecules of it. But we also produce what's called reducing equivalents. So this is a molecule NADH. Okay, don't worry about its full name. And the first half of glycolysis requires energy, and the second half is the payoff stage where we generate that net 2 ATP. So really what I want you to look, the overall reaction that happens is we have a glucose molecule, we have 2 of these little NADs, we have 2 ADPs and 2 phosphates. The 2 ADPs and the 2 phosphates form 2 ATP, so adenosine diphosphate goes to form adenosine triphosphate. Our 2 NADs go on to form NADHs. They just have hydrogen ions added. Okay, and that will be used elsewhere to make more ATP. And our glucose, which has got 6 carbons, goes on to form pyruvate, but 2 of them. Each pyruvate has got 3 carbons. So how is it all regulated? Well, it's all very, very complex, okay? And we're just going to really pick up on the key points. So if we're looking at the reaction that's actually happening from glucose all the way down to pyruvate, you can see we go glucose, we eventually go down to fructose 6-phosphate, then fructose 1,6-bisphosphate, before going on to pyruvate. And of the enzymes, because there are enzymes doing each of these little steps, the most important one that's regulated is this little one. The one that goes from fructose 6-phosphate to fructose 1,6-bisphosphate. And that is called phosphofructokinase. Okay, and it's stimulated by some things and it's inhibited by others. So for example, it's inhibited by ATP. Okay, so the more ATP you have means that you don't really need to be metabolizing your glucose. Okay, so ATP inhibits it because you don't need to make more ATP. So why stimulate this pathway? So you inhibit that pathway. It's also inhibited by citrate. Now citrate is the first product of the citric acid cycle, the place where pyruvate goes afterwards. Okay, so if you get a build-up of citrate, it means we don't need this pathway to be going on. We've got plenty of intermediates that are going on to produce energy. So citrate also inhibits it. A low pH also inhibits this pathway, as does glucagon, which is a hormone. So a hormone that goes on to increase our blood glucose levels. Okay, so that inhibits the breakdown of glucose because really we want to be making more glucose. We want more glucose going into the blood, not less. So that's really what's happening to do. Well, why is pH doing it? Well, really, pH helps to reduce glycolysis in an anaerobic environment, just really to prevent too much lactic acid buildup, because lactic acid is an acid and acids are toxic. So what about what stimulates it? Well, of course, if you're running out of ATP in your cell, you're building up AMP, adenosine monophosphate, and ADP, adenosine diphosphate. And we need ATP, adenosine triphosphate. So basically, if you've got lots of AMP and ADP, that stimulates it because it's kind of going, come on, let's make more ATP, let's make more ATP. And likewise, adrenaline and insulin, they also work to stimulate this pathway. Okay, because insulin, of course, is kind of going, that's you just had a meal, you've got lots of glucose, and it's saying, well, let's use that glucose. I don't want to use anything else. I don't want to use my stores of fatty acids, for example. I want to use the glucose that I've already got. So all in all, this entire process is ensuring that the glycolytic activity, so that's glycolysis, the activity of glycolysis, increases when ATP is needed and reduces when all the intermediates are abundant. So in other words, where we're able to produce lots and lots of ATP. So what about anaerobic glycolysis? Well, of course, this occurs when oxygen levels are low. And what happens is in this area, so here we go again, glucose goes down to pyruvate. So this is that normal pathway that we talked about. So glycolysis, ADP to ATP, two of them, two NADs to two NADHs, we form our pyruvate. But when it's anaerobic glycolysis, we're forming lactate, okay, or lactic acid. And actually we don't get any net production of NADH during this process because NADH is used as a product or a byproduct or as an intermediate to produce our lactate. So in other words, glucose to pyruvate produces 2-NADH, pyruvate to lactate uses those NADHs again. So it all kind of gets recycled in that pathway. So in other words, what's happening there, the two molecules of pyruvate that are produced from one glucose molecule, one molecule of NADH is used to produce one lactate from one pyruvate, therefore we're using two NADHs per anaerobic cycle. And in the aerobic pathway, the pyruvate enters the mitochondria, converted to acetyl-CoA, where it enters the citric acid cycle, and we get the NADH produced during aerobic glycolysis, so this stuff over here, also enters the mitochondria and gets converted through the electron transport chain that we'll talk about later into ATP. So when does lactic acid build up? Oops. So there's that same diagram. Clinically, of course, the most important cause is impairment of oxidative metabolism. So for example, during respiratory failure, for example, if there is insufficient oxygen transport, so you're not transporting oxygen efficiently enough, or if there are toxins preventing oxidative phosphorylation in mitochondria, so that just means producing more and more ATP, or we're not producing enough ATP, so therefore we're preventing oxidative phosphorylation in the electron transport chain, so we're having to produce ATP from somewhere else. But of course, during intense exercise, it also builds up in our skeletal muscle. And without oxygen, mitochondria cannot oxidize NADH to NAD. So in other words, remember what's happening. We produce lots of NADH, this lovely little molecule that can go on to produce ATP. But if we haven't got any oxygen, we can't oxidize that NADH and therefore go on to produce ATP. And that accumulating NADH makes the lactate dehydrogenase reaction, so that's the one that produces the lactate, irreversible in the direction of lactate formation. And therefore lactic acid builds up. We have a lower pH that alters protein function, causing cell damage. But really like we've said, the most clinically important times this will happen is in respiratory failure, insufficient oxygen transport, and when you have certain toxins building up, preventing the electron transport chain working sufficiently within the mitochondria. So as we've talked about, when we break down glucose into pyruvate, but also fatty acids, amino acid metabolism, they all go on to produce this molecule, acetyl-CoA. So here's your pyruvate, okay, that comes in, so pyruvate from glucose, remember, that comes in to produce acetyl-CoA. Remembering, 3 carbons there. Acetyl-CoA has only got 2 carbons, so we lose our first carbon as carbon dioxide at this stage. And lo and behold, we produce NADH. Very handy. But of course, when we break down our amino acids by catabolism and our fatty acids by beta oxidation, we also form acetyl-CoA. So this molecule, acetyl-CoA, is a great little intermediate or molecule that is forming within the mitochondria. And as I said, that carbon dioxide is produced during the production of acetyl-CoA from pyruvate. So there it was, your 3 carbons entering the mitochondria to form your acetyl-CoA, and we lose a CO2. And CO2, of course, is released as a waste product. I guess at this point I should also mention that carbon dioxide produced from pyruvate, it's not actually within the citric acid cycle. It's actually happened before the citric acid cycle. Okay, so it's not a byproduct of the citric acid cycle, it's a byproduct of pyruvate conversion into acetyl-CoA. So acetyl-CoA is basically the entry point into the citric acid cycle. Like I said, CO2 is being produced beforehand. And so what you can see here is the citric acid cycle. Now it looks really, really complex, okay? But what I really want you to look at, first of all, is here is acetyl-CoA. This is the starting off point. This is what our fatty acids have been broken down to form. This is what our amino acids have been broken down to form. This is what our pyruvate, which has come from glucose, has been broken down to form. All of which have entered into the mitochondria. And this is the starting point of the citric acid cycle. Okay, so this is the starting point. This is where we're at this point. This acetyl-CoA combines with another molecule that's hanging around in the mitochondria called oxaloacetate. Okay, it's just got 4 carbons. So each of these little circles represents a carbon. So you can see 4 carbons from oxaloacetate, and it's a cycle, so it goes wrong way. It goes round and round. Okay, we've got 4 carbons. We've got 2 carbons from acetyl-CoA. So we now know they combine, and we're going to have 6 carbons. Okay. And 6 carbons, the first molecule is citrate. So that's what formed. If you remember, citrate was involved in the regulation of glycolysis. So the more of this citrate that you form, you kind of go, oh, I don't need glycolysis to be going on. So it inhibits glycolysis, if you remember. Okay, so that's where that came from. We then start going around this pathway, okay, and let's not worry too much. And they're not named in this, which is quite handy. So we basically lose a carbon as carbon dioxide and form an NADH. We do it again, release a CO2 and form an NADH. Okay, so at each point we've lost 1 carbon dioxide there, we've lost another carbon dioxide there. We then go through another few little pathways and we form GDP to GTP. It's basically an equivalent to ATP. So we're thinking, yeah, we're getting some energy. We then produce FAD to FADH2. That's an equivalent of NADH. So we're producing some reducing equivalents. We go round the pathway again and we produce another NADH and we're back to oxaloacetate. Really do not worry about what any of these intermediates really are. If you can just understand what's happening in the citric acid cycle, we're starting oxaloacetate and acetyl-CoA. We form our citrate, which is important, and then we just produce lots of reducing molecules, NADH, NADH, FADH, NADH, release a couple of carbon dioxides, and come back to our original molecule. So I want you to have a little look at this, okay? And I want you to look how many molecules of ATP, GTP, which is basically like ATP, NADH and FADH, and CO2 are produced in any one round of the citric acid cycle. So just one round, just starting with 1 acetyl-CoA, okay, and 1 oxaloacetate. How many are formed? So first of all, have a little look around there. How many ATPs have you seen? Hopefully you said none, because I can't see any. How many GTPs can you see? Boo! 1. We've got 1 GTP. Sorry for the sound effects. How many NADHs? 1, 2, 3. How many FADHs? 1. And how many CO2s? 1, 2. And so there it is. So per citric acid cycle round, from 1 acetyl-CoA, we basically produce no ATPs, 1 GTP, 3 NADHs, 1 FADH, and 2 CO2s. So how many of each of those are produced per glucose molecule? Only thinking about the citric acid cycle, not thinking about glycolysis. Okay, I just want you to think about the citric acid cycle. How many of each of those are there? Okay, so looking around here, so remembering what's come in. Pyruvate has been up here. This is from your glucose. Pyruvate forms your acetyl-CoA and loses a CO2. So basically, one glucose forms two acetyl-CoAs. And as we've now got two acetyl-CoAs, basically everything doubles. So no ATPs, two GTPs, 6 NADHs, 2 FADHs, and 4 CO2 molecules. And just like glycolysis, the citric acid cycle is very, very tightly regulated. Okay, and it's tightly regulated by the product of the cycle. Okay, and so this is that cycle again. We've got the acetyl-CoA. These are some of the intermediates. I'm not too worried about you remembering the names of the intermediates. But really what I want you to think about is if we're thinking about these four molecules, ATP, ADP, NADH, and acetyl-CoA, which of those are going to stimulate and which ones are going to inhibit the pathway, the citric acid pathway? Like I said, don't worry about these intermediates. It's really just not important. But what I want you to understand is the concept of if these build up, will they stimulate or inhibit the pathway? Okay, so hopefully you got this. So, what inhibits the pathway? Well, obviously ATP and NADH, because you're producing lots and lots, and again this one, but you're producing lots and lots of ATP and NADH, or ATP equivalents in this pathway. Okay, so because remember, if you have this pathway, you produce NADH, and the NADH will go on to form lots more ATP. So if you have lots of ATP and you have lots of NADH, you don't need to keep stimulating this pathway, so you inhibit it. And that's what NADH and ATP do. And of course, if you haven't got enough ATP in your cell, then the cell kind of goes, hey, I want more ATP. So ADP stimulates it, because if you've got lots of ADP, it means you haven't got enough ATP, and it wants to make more ATP. So ADP stimulates this pathway, which means we produce more NADH, which means we can go on to produce more ATP. Okay, so we're going to do a quick energy check. What's actually happening? What have we produced so far? So, glycolysis. Here we go. Glucose takes place in the cytoplasm, remember. Glucose to pyruvate, or two pyruvate molecules. We have two NADHs and two ATPs at this stage. The pyruvate is converted to acetyl-CoA. We lose a couple of CO2s because the acetyl-CoA is 2 carbons, pyruvate is 3. But we produce another couple of NADHs, marvellous. That acetyl-CoA enters the citric acid cycle, or the Krebs cycle, or the TCA cycle. They're all the same thing. And in that process, overall, it produces 6 NADHs. It produces 2 FADHs. It produces 2 GTPs, which are the equivalent of ATP. And it produces 4 CO2s. So basically, if you look at this, we've gone glucose, C6, 6 carbons. Lost 2 of them there, lost 4 of them there. We've metabolized an entire glucose molecule. So therefore, we've got 4 ATPs, 2 from glucose and 2 from GTP. And we've got a lot of reducing equivalents, okay, which go on to produce more ATP. We've got 10 NADHs, 2, 4, 10. We've got 2 FADHs, there they are. Okay, so we've got 4 ATPs and we've got 12 reducing equivalents, which will go on to produce lots and lots more ATP. So the next step that happens is the electron transport chain. And so what happens in this is the electron transport chain forms a proton gradient in the mitochondrial membrane. And it's very important because gradients allow us to then go on to produce energy as molecules flow down their gradient. So what happens? Looks very complex, but really, hopefully it isn't too complex. So here we have the mitochondrial membrane, okay? And what you can see, it's got a dual bilayer. So there's one bilayer there, bilayer, because bilayers, one layer, that's a bilayer, and there's a second bilayer. And in between there is an intermembrane space, okay? And in that in the inner bilayer, we've got all these little molecules or complexes, protein complexes they're called, okay? And what they do is basically they work to generate or pump hydrogens into this intermembrane space. So really they're using the energy from NADH to convert the NADH to NAD. Electrons start flowing through this whole pathway and hydrogen ions pump into this intermembrane space. So if you look at it, NADH converted to NAD+, FADH converted to FAD. Okay, and this moves all our electrons along, and it also causes this hydrogen ions to move in to the intermembranes. So what is actually happening? Well, we can see that NADH, as I've said, is coming in at this first complex. Okay, so it's coming in complex 1. FADH is coming in at complex 2. Just so you know the names, this one's called NADH dehydrogenase because it's working on NADH. This one's called succinate dehydrogenase. This third one, complex 3, is cytochrome C reductase. Fourth one is cytochrome C oxidase. And the last one, complex 5, is ATP synthase because it goes on to make ATP. Don't worry too much about the names. Complex 1, 2, 3, 4, 5 is absolutely ample. Okay, but what's actually happening in this whole pathway? So this is where we're starting off. And you can see that as we start to do this whole bit of work, the electrons are passed down the chain. And as they pass down the chain, they release energy in the process. And as they are releasing energy, protons, your H+ ions, are pumped against their gradients. They're basically, there's more hydrogens here, of course, and you're pumping them, there's more hydrogens here, so you're pumping them against the gradient. So they're going into the intermembrane space against the gradient. And then when you've got a nice build-up of that gradient through complex 5, the hydrogen ions now flow from a high hydrogen ion concentration to a low hydrogen ion concentration down its gradient through this ATP synthase, where we go on to make ATP. So this is how we generate ATP. We're using all these NADHs and FADHs that were produced during the citric acid cycle to generate a proton gradient, which then means we can make ATP. At the same time, of course, as you can see from this, we split an oxygen atom or an oxygen molecule. So an oxygen molecule is split into an atom, joins with hydrogens, and we form water as a byproduct. So as I've said, in this entire pathway, protons flow down their electrochemical gradient through ATP synthase, leading to the synthesis of ATP. So here it is, it's just gone the other way. Okay, so there's 1, 2, 3, 4, and there is our number 5. So the hydrogen ions go through this ATP synthase, go on to form ATP. This is called chemiosmosis. Okay, it's like osmosis, like a gradient, except it's chemical. It's hydrogen ions that are moving. So what I'd like you to really do right now is look at this diagram and just describe exactly what is happening. If you're with someone, talk to the person next to you. If you're not with someone, just press pause and have a think about and talk to yourself, if you like, about what's happening in this process. Okay, so hopefully you've done that. And at the end of the day, what happens? Through the whole metabolism of a single glucose molecule, we produce about 32 molecules of ATP. Okay, most of them are coming through the electron transport chain, which is this whole process, but some of them are coming through glycolysis, for example. And we're getting a couple of GTPs, which is the equivalent to ATP, through the citric acid cycle. But most of them are coming through the electron transport chain. But what happens if this electron transport chain doesn't work? It can be uncoupled. And uncoupling of this electron transport chain basically means it's not going to work anymore because it's been uncoupled. But when it becomes uncoupled, it means we'll get a reduction in the amount of ATP that's being produced. And if we're reducing the amount of ATP that's being produced, it means we'll produce more heat because less ATP equals more heat. So basically this occurs, the uncoupling occurs when uncoupling proteins, that's these UCPs that you can see on the right hand side here, provide an additional route for proton transfer into the matrix. So remember what happened, the electron transfer chain was breaking down these reducing equivalents and transporting hydrogen ions into the intermembrane space. Now if it's coupled, okay, it goes through ATP synthase and we produce lots of ATP. But sometimes it can become uncoupled. And instead of producing ATP, the hydrogen ions just pass back down and we produce heat as a byproduct. Okay, so I'd like you to think now, without pressing the next buttons or anything else, I want you to just press pause and think, when might that happen? Okay, I'll give you a clinical example in a second, but I also want you to think about when it might happen at a different point. When does it happen? Well, actually, it's a common occurrence in brown adipose tissue, which babies have. Okay, babies have lots of brown adipose tissue. It's more metabolically active. And in hibernating animals to keep them warm. So instead of producing ATP, animals are hibernating. They don't need all this ATP. It's there to keep them warm. And brown adipose tissue, think about in infants who don't have the shiver reflex. This brown adipose tissue is a way of keeping the baby warm. It's uncoupled and we produce more. It goes through these uncoupling proteins, producing heat as a byproduct. But also, just as another one, salicylate or aspirin poisoning allows protons to diffuse back into the matrix as well, producing heat. So aspirin poisoning leads to excess heat production and by its very nature, less ATP production as well. Okay, so we've gone through glycolysis, we've gone through the citric acid cycle, we've gone through the electron transport chain. I fully understand it's quite a hard topic for most people. Hopefully you'll understand most of what we've gone through today. But here you go, I'm going to give you a little question for you to have a think about. So obviously, as usual, press pause. I want you just to have a look. Which of the following are correct? So one or more may be correct. So 1, 2, 3, 4 may be correct. None may be correct. Just true or false in each case, basically. So I want you to have a look at each one. Press pause. Okay, which ones of the following are correct regarding the electron transport chain? Pesticides such as rotenone, an inhibitor of one of the early electron transport chain complexes, will increase the size of the proton gradient. So basically you're inhibiting one of the complexes involved in the electron transport chain. Do we get an increase in the size of the proton gradient? True or false? Obviously it's false. You'll get a much smaller proton gradient. You're inhibiting the complex. Therefore you can't pump the hydrogen ions into the intermembrane space. Therefore you form a smaller gradient and therefore less ATP. Next one. Cyanide and carbon monoxide both bind to complex 4. This will prevent electron transfer to oxygen. Correct. This will uncouple the complexes, meaning that ATP synthase basically won't work. Okay, we're uncoupling the complexes because they all need to remain coupled together. Hence, uncoupling and coupling. 1, 2, 3, 4, 5, all working together. Number 3. Inherited mitochondrial disorders should be suspected in patients with muscle or central nervous system disease and involvement of 2 additional systems. Very hard question for you, but I just want you to have a little think about it. True or false? Why would I even put it in there? It's actually true. Okay, I've really put it in there just to make you aware of it, that some of the inherited mitochondrial disorders should be suspected where we have issues with muscle or CNS disease and 2 additional systems. It doesn't mean that it is an inherited mitochondrial disorder, but it's been put in there so that you are aware that it may be an inherited mitochondrial disorder. Okay, so it's just something to have in your brain, in place there. Next one. The increased brown fat evident in infants help them stay warm in the absence of the shivering reflex. Well, hopefully you got that one right because I talked about it earlier. That's correct. So infants have a much higher amount of brown fat. They produce less ATP from these fat cells, but they do produce more heat, which means they can stay warm in the absence of the shiver reflex. And it's quite interesting, actually, that there are studies looking into the upregulation of the uncoupling proteins. So you remember those uncoupling proteins? People are trying to look into how we can upregulate those to help treat obesity. Because then what will happen if you can uncouple them in your adipose tissue, you can start to lose more energy as heat rather than producing ATP to do stuff. And therefore, as you're getting less ATP is being produced per molecule of glucose or fat or whatever it might be, you're basically going to potentially lose weight. Okay. So the third part that we're going to look at is we're going to start looking at hormones. And really, this is looking at hormonal regulation of insulin and glucagon. We're going to mainly talk about, but actually, there are lots of hormones that are regulating fuel metabolism. Okay, those are 2 that you probably know about, but actually there are 5 that are involved in fuel metabolism, and we will touch on all of those at this point. So when we're talking about the hormonal control of fuel metabolism, we're really thinking about blood glucose levels or blood amino acids or lipid fatty acid levels, but really we focus all the time on glucose. And we maintain blood glucose levels within very narrow limits, and that's critical for our nervous system and our brain function. Remember, our brain must be using glucose at all times, even during starvation. 50% of the energy it is using is coming from glucose. And our fasting blood glucose levels are about 3.5 to 5.5 millimoles per litre. These are numbers you'll get to know very well over time. So why do we need blood glucose? Well, of course, energy, ATP production, in particular for the brain, you're thinking action potentials. And as I've said, the brain requires glucose at all times, even during starvation when other tissues are using fats as an energy source. And at rest, it's 2% of your body weight, but it's using 75% of your body's glucose. Remember, your red blood cells, they only use glucose as an energy source as well, so they work anaerobically. But which hormones regulate blood glucose levels? So we're going to focus on 2. So I want you to think, first of all, which hormones reduce blood sugar levels? Hopefully you said insulin. In that case, you'd be right. Now I want you to tell me which hormones, and there's an S on the end of that, increase blood sugar levels. So glucagon, obviously, I'm presuming you got that right. But there are 3 more. There are 3 other hormones that increase our blood sugar levels. You can think of it as like this. If you have a low blood sugar levels, that's stressful on the body. So what are the stress hormones? Adrenaline and cortisol. So those 2 hormones also increase blood sugar levels. Okay, so adrenaline is our short-term stress hormone, cortisol, our long-term stress hormone. But that's 4 we've got so far. Insulin reduces it, glucagon increases, adrenaline increases, cortisol increases blood sugar. There's one other hormone that is produced that increases blood sugar levels. Very important actually at birth. Okay, it helps to increase blood sugar levels straight after birth. And that's growth hormone. Okay, so growth hormone also increases your blood sugar levels. So there they go. Insulin, glucagon, adrenaline, cortisol, and growth hormone. They're the only 5. Okay, some people say incorrectly in exams, right, thyroid hormone. Please do not put it there. It is wrong. Insulin reduces it. Glucagon, adrenaline, cortisol, growth hormone increase our blood sugar levels. So how is this whole process regulated? How are the hormones actually working? What's going on? So presuming you've just had a meal, shortly after a meal, your blood sugar levels start to rise. And where does that food come from? So here's your small intestine all around here, okay? And what happens to, so here's your small intestine. And when you start to process that, you absorb it through your epithelial cells and into your hepatic portal vein. So nutrients, amino acids and carbohydrates go straight to the liver. That's the first place they go to. And we're really thinking about sugar, okay? So glucose is absorbed through the GI tract, enters the hepatic portal vein, goes to the liver. And in the liver, basically, it takes what it wants. So the liver takes glucose through the GLUT2 transporter and it uses it as necessary. And it'll store glucose as glycogen as well. But the whole point is, it's not regulated by any hormone, this. The liver just takes whatever glucose it wants through the GLUT2 transporter and stores it as glycogen. But several hours after a meal, we're starting to run out of blood glucose levels. So the blood glucose is being reduced. So now we need to get more glucose into the blood. And this is where glycogenolysis. So we start to break down the glycogen that was made in our liver. We break it down to glucose, and that glucose enters our systemic circulation to be delivered to the tissues that need it, primarily the brain, of course. We also stimulate gluconeogenesis. So that's the synthesis of glucose from amino acids, for example, again, in the liver. And this glucose is released from the liver to maintain normal blood sugar levels. And as we've talked about, much of this is dependent on the hormones. So insulin it promotes glucose use and storage. So basically after a meal, insulin causes the glucose that is being delivered to the liver to be converted into glycogen. Okay? And glucagon, cortisol, growth hormone, and adrenaline all serve to increase our blood sugar levels. They help to break down the glycogen in the liver into glucose, which can go to our tissues that require it. So in other words, if we're thinking about anabolism, it's insulin. Insulin is signaling the fed state. It switches on the pathways for cellular uptake and storage and switches off the pathways for breakdown. We don't need to break down our stores of energy, our glycogen and our triglycerides if we've got plenty of food coming in. So what's actually happening? Insulin is triggering the uptake of fuel substrates into cells, specifically skeletal muscle and adipose tissue. Okay, and that is through what are called GLUT4 transporters. So they are the insulin-dependent glucose transporters. It also stimulates the storage of fuels. So we store lipids, triglycerides in our adipose tissue. We store glycogen in our liver and skeletal muscle. That's all being stimulated by insulin. It stimulates the biosynthesis of proteins and DNA. And it stimulates glucose utilization. Okay, so basically we want to use the glucose that's coming in. As we're storing it, we're also wanting to use it. We don't want to use our stores of energy when we've got energy coming in from a meal. So glucose utilization, either as storage molecules or as biosynthetic molecules, or just for production of ATP, means blood glucose levels fall to normal, which then causes insulin secretion to reduce. So I want you to look at this table. And I want you to tell me, in each case, is it a yes or a no? So what does insulin actually do? So it's released following a meal and therefore serves to store energy. But what's happening? So do we get increased glucose uptake into cells via the GLUT4 transporter? Do we increase glycogenesis? Does it inhibit glycogenolysis? Does it increase lipogenesis, so lipid synthesis? Does it inhibit gluconeogenesis? And does it increase amino acid uptake and protein synthesis? Okay, some of you may know the answer to that because there are plenty of bodybuilders who take insulin for that very purpose. So I've kind of given you a little clue on that one. So again, press pause, work your way through the table. Okay, so increased glucose uptake into cells. So does insulin stimulate increased glucose uptake into skeletal muscle via GLUT4, the insulin-dependent GLUT4 molecule, transporter rather? The answer is yes. What about adipose tissue? Yes. What about the liver? No. Remember what happens there? Following a meal, all the nutrients, the glucose goes straight to the liver and the liver just takes what it likes through a different transporter, the GLUT2 transporter. The GLUT2 transporter is also used in the pancreas. The GLUT3 is used in the brain, and there are other systems that use different glucose transporters. So GLUT4 is only really skeletal muscle and adipose tissue. So you're only getting insulin-dependent glucose uptake into two main cell types. Do we get an increase in glycogenesis in skeletal muscle? So glycogen synthesis. Yes, we do. What about in adipose tissue? No, because we don't have glycogen stored there. What about in the liver? Yes, because we do have glycogen stored there. Does it decrease glycogen breakdown? Skeletal muscle? Yes. It's making it, so obviously it's going to stop it being broken down at the same time. What about in the adipose tissue? No, because there isn't any glycogen. And liver? Yes. It's stimulating it to be made, so it's decreasing it being broken down at the same time. Does it increase lipogenesis? No, I've written no there, but there are small stores of triglycerides in your skeletal muscle, so just be aware of that. But I've written no because it's not a major role. Adipose tissue. Yes, of course, it stimulates fat synthesis in fat cells so we can store that fat. What about in the liver? Yes, it stimulates fat synthesis in the liver, and that fat or those triglycerides can then get transported to the adipose tissue where they can get stored. Does it decrease gluconeogenesis? So does it decrease glucose synthesis? So you've got to think, following a meal, do you need to start making glucose? The answer is no. But where does gluconeogenesis take place? So do we decrease gluconeogenesis in the skeletal muscle? No, because gluconeogenesis doesn't take place there. What about in adipose? No, because it doesn't take place there. What about in the liver? Yes, it decreases glucose synthesis from things like amino acids because we've got plenty of glucose already. We don't need to be making more. So does it increase amino acid uptake and protein synthesis in our skeletal muscle? The answer is yes. Okay, and like I said, that's a reason that some bodybuilders take insulin to try and build up or synthesize take up amino acids and build more proteins. Adipose tissue, no, we don't really go on to make a lot of proteins in there. Liver, yes, we do, okay, because it makes a whole range of proteins for the body. And so that's what insulin does. So I'll quickly get rid of that again. So that's what insulin does, very crudely, okay, that's its function. And remember what happens in diabetes mellitus. In type 1 diabetes, you have a lack of insulin. And in type 2, you have insulin resistance. So basically, in diabetes mellitus, to varying degrees, the opposite of the yeses above occur. So if you have diabetes mellitus and it's poorly controlled, that becomes no, 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 etc. So basically, the whole table is a no, okay, in diabetes mellitus, poorly controlled. So if we're looking at a summary of fuel metabolism, of what's really happening, you can see what we have is if we have, let's start with it there, a high blood sugar, so up at the top. High blood sugar promotes insulin release. That acts on the beta cells of the pancreas, and the beta cells of the pancreas then go on to stimulate glucose uptake in your adipose tissue and your skeletal muscle. And therefore in the liver, as well as that, it stimulates glycogen formation. So we're forming glycogen there, and we're stimulating glycogen in our skeletal muscle as well. And all of that starts to lower our blood sugar levels. If our blood sugar levels are too low, it promotes glucagon release from the alpha cells of the pancreas, and that stimulates glycogen breakdown in the liver to glucose, and that glucose enters the blood, which raises our blood sugar. So that's a summary of those two little hormones, some of the features. So of course, glucagon does lots of things. It breaks down glycogen, as we've just talked about there. It breaks down lipids. You remember, you've got a low blood sugar, it's kind of going, oh, I haven't got enough energy, so it'll break down lipids, so that we've got those as an energy source. It'll make ketones. It'll go, yeah, I'll make some ketones, we need a bit more energy. And it'll stimulate gluconeogenesis. It'll make glucose from your amino acids. Cortisol also increases our blood sugar levels. It also stimulates glucose synthesis. It actually stimulates the synthesis of glycogen. Basically, if you think it's your long-term stress hormone, so I want you to think if you're a stressed or diseased or injured animal is probably a better example. You might not feed for a while. So it's building up your glycogen stores so that if you are attacked by another animal, you've got some energy for that fight or flight response. Really, that's a fundamental reason. It stimulates proteolysis, breakdown of proteins, so that the amino acids can go on to make glucose during gluconeogenesis. And again, it stimulates lipolysis, breaking down your lipids. And adrenaline and growth hormone both stimulate glycogen breakdown, and they both stimulate lipolysis. So all of these molecules are basically increasing our blood sugar levels, okay? So we've got gluconeogenesis, glycogenolysis from glucagon. Cortisol, gluconeogenesis, and we've got adrenaline, glycogenolysis, and growth hormone, glycogenolysis. So all of them are increasing our blood sugar levels, which means if you have a tumor, for example, that secretes too much of glucagon, cortisol, adrenaline, growth hormone, or any other cause of an increase in those hormones, you're likely to become diabetic because your blood sugar levels rise. So, as I mentioned earlier, glucagon is released from our pancreatic alpha cells. They're in our pancreas. Stimulated by low blood sugar levels. Hypo is low, hyper is high, hypoglycemia, emia in the blood, so low blood sugar levels. It enhances amino acid uptake into the liver for gluconeogenesis. It stimulates lipolysis in adipose tissue, glycogenolysis in the liver, gluconeogenesis in the liver, and ketogenesis in the liver. So it's doing a lot of things in the liver, all of which serve to increase blood sugar, blood ketones, blood fatty acids, all of which can be used as energy sources. So in other words, if we have a decrease in our plasma glucose levels and a rise in our amino acid levels, stimulates glucagon from the alpha cells of the pancreas, stimulates adipose tissue to break down fats to form fatty acids. And fats are used by tissues, which helps to spare glucose. We're not using glucose in all of those tissues. Your heart can use your fat. Your skeletal muscle can use the fats. It doesn't have to use glucose. It also stimulates glycogen breakdown and glucose synthesis in the liver, increasing our blood glucose levels. And as your blood glucose levels rise, so does your insulin, and therefore less glucagon is released. And of course, just remember a couple of other things. Cortisol is what stimulates proteolysis, breakdown of proteins. Again, released following in between meals to have enough glucose. And the amino acids can be used for gluconeogenesis, so glucose synthesis. But cortisol also stimulates glycogenesis and lipolysis as well. So remember what we've talked about. We've talked about gluconeogenesis being the synthesis of glucose from non-carbohydrate sources. So I've talked about that a lot, and I've talked about how it's coming from amino acids. But just to give you a little bit more information on that, your amino acids, your glycerol and your lactic acid, they're all sources for gluconeogenesis. Okay, and this is your citric acid cycle, your Krebs cycle. You don't need to remember this. Okay, you don't need to remember it. But the important point for this, if I want you to be looking at it, is you've got all these different amino acids at different places, and they all enter the citric acid cycle at different points. Okay, and then when they enter the citric acid cycle, gluconeogenesis, which is the important bit, comes off from oxaloacetate and goes on to form glucose up there. Okay, so that's how it undergoes gluconeogenesis. The amino acids all come in, they all get processed through the citric acid cycle, and we go on to stimulate gluconeogenesis. Okay, and therefore glucose synthesis. And gluconeogenesis provides a significant proportion of the glucose found in your plasma, up to 50%. Okay, so gluconeogenesis is a process that is always going on, and so therefore you're always having to break down some amino acids to stimulate it. So, a question for you, a reminder question: can you remember which hormone or hormones stimulate gluconeogenesis, and what main tissue or tissues does it take place in? I'll give you a clue. 2 hormones stimulate gluconeogenesis. What are they? Cortisol and glucagon. They both stimulate gluconeogenesis, and the main tissue for gluconeogenesis is, of course, the liver. So how is all of this being regulated in the starvation phase, if you want to think of that? Let's think about it like this. So, of course, the first phase whenever you have food is your absorptive phase. Okay, and this is the amount of glucose used per hour against grams per hour against the time scale. And so you've just eaten a meal with lots of glucose, and basically you're absorbing it. This is the stuff you've eaten in, hence why it says exogenous. And basically this is the source of your glucose following a meal. And really it lasts up to about 4 hours. You can see that there. And after about 4 hours, we're hitting the post-absorptive stage. Okay, so that's what you're seeing at this point. And in the post-absorptive stage, this is the short term. And this is, as you can see, we're mainly using glycogen as an energy source. This is liver glycogen, remember, and not skeletal muscle glycogen. Liver glycogen is being broken down to glucose, which enters the blood. It's regulated mainly by glucagon during this period, also to a lesser extent cortisol, and it's mobilizing liver glycogen to form glucose, and our triglycerides are being broken down into fatty acids. But the amount of glycogen we have in our liver is very limited. It's only about 100 grams. Okay, so it's not a huge amount of energy. It's about 400 calories. So the phase is quite short, and you can see that there. The phase is quite short. By 16 hours, an overnight fast, if you like, we've lost 50% of our glucose, of our glycogen stored in our liver, rather. So then we hit that early starvation phase. We've hit this point where we've used, half of our glycogen, and we've really done nothing during that entire time, but yet we've used half our glycogen. And so during this period, we need this amount of energy, or glucose. So we're getting it now from gluconeogenesis. So at 16 hours, we're getting half of our glucose from gluconeogenesis, from amino acids, and half of it from glycogen. So in this early starvation phase, we're really now being regulated by glucagon, but also much more significantly by cortisol, because cortisol is breaking down the proteins into amino acids, which can be used for gluconeogenesis. So now these 2 hormones together are stimulating gluconeogenesis using non-carbohydrate materials, such as amino acids, and they're both also stimulating lipolysis for those other tissues that don't require glucose. But we couldn't maintain this early starvation phase. We've run out of glycogen, okay? And so we're having to use amino acids only at this point. That's not sustainable, okay? Because it would lead to muscle wasting, so it can't be maintained. So in that intermediate and prolonged starvation, we start to produce ketones. And this is regulated by cortisol and glucagon. And this is where our tissues have really shifted to fatty acids as their main energy source. Those tissues that can use fatty acids are using fatty acids. The muscles are preferentially using fatty acids over glucose. The brain still needs about 50% of its energy to be coming from glucose, so we still got to have some gluconeogenesis going on. There it is, we're still breaking down our muscle. But it also can use ketones. Okay, so it can use the ketones which have been made from from fats. Remembering, of course, red blood cells also need to use glucose as an energy source. And so, if we're thinking about the rough proportions, the brain uses about 30% of its energy as ketones at day 3 and about 50% during prolonged starvation. So finally, I've got a question for you to think about. So press pause and have a little read of the following and say true or false in each case. Okay, number 1. Ketone bodies or keto acids, the main ones being acetoacetate and beta-hydroxybutyrate, are acids produced from acetyl-CoA in the liver during starvation. Yes, that is correct. Remember, the ketones can be used as an energy source, in particular in the brain. Ketone bodies are converted to acetyl-CoA in the brain during starvation, where they could be used in the citric acid cycle to provide energy. Also correct. So when the ketones are transported into the cells in your brain, so into your neurons, they're converted back into acetyl-CoA, which is the starting point of the citric acid cycle, where we start to produce NADH and ATP and GTP. Only the brain can use ketones as an energy source. This is incorrect. Many tissues can use them, in particular the brain, skeletal muscle, and heart. Type 1 diabetics are susceptible to ketoacidosis, whilst it is rare in type 2 diabetics. Hard question for you if we haven't covered diabetes yet. It's actually correct, because insulin inhibits ketogenesis. So if you have a lack of insulin, plus the presence of glucagon, this leads to ketone formation. It doesn't happen in type 2 diabetics because they have insulin resistance, remember. Whereas type 1 diabetics, if it's poorly controlled, have a lack of insulin. So in type 2 diabetes, it's rare because they have insulin resistance, but they have enough insulin action to prevent ketoacidosis. In other words, their bodies do not become acidotic. Ketoacidosis is a metabolic acidosis, and compensation is through respiratory hyperventilation. So ketoacidosis is a metabolic acidosis because it's being produced during metabolism, so that bit is correct. How does the body compensate? How does it get rid of that acid? Is it through respiratory hyperventilation? Well, yes, it is. If you hyperventilate, you release or you lose more carbon dioxide, and I want you to think of carbon dioxide as an acid. So in other words, CO2 is an acid, so hyperventilation is the respiratory compensation for a metabolic acidosis.